So before we start these afternoon activities and a few of the activities online, I just wanted to show you a video that I recorded last year in Mongolia while sampling some of the hot spring, because we were discussing the biofilm, right? So are we all, by the way? 1, 2, 3, 4, 5, 6, 7. No, there's a lot of people missing. Where are the others? Vuoi fare un fischio fuori? Your 2 neighbors are missing. No, they were outside chatting. So I don't know how the resolution will be in the big video, but you'll see it in a second. This is a microbial biofilm that contains calcite, precipitated calcite, okay? It's actively precipitated calcite in the area. Take a look. That's me with my knife. And it's not even one of the thickest we found around. Take a look. It's like 1.5 centimeter thick layer of things. And you can see with my knife how deep it goes. Look at that. Yeah, microbial brownies, exactly. So that said, let's start. So this afternoon, we will get you play a little bit with Winogradsky columns. Brief introduction. You remember this, right? We discussed metabolism. Metabolism as redox chemistry. Typically, you have an electron donor, an electron acceptor. Microbes do the redox couple, but they do splitting it into different space and time. So at the top, for example, we have thiosulfate oxidation, and then at the bottom, we couple it with oxygen. There is a completely different enzymes, okay? So 2 enzymes, one is gonna break down, take electrons somewhere, like from hydrogen, hydrogenases, and then those electrons will be transferred to another enzyme that will dump them on a secondary electron acceptor. And we also said that the role of cell is actually to couple different metabolism, to couple different redox and acceptor, to have crossing between the different biogeochemical cycle. So a cell might be using hydrogen and oxygen for energy, but it still needs carbon, needs nitrogen, needs phosphorus, needs a number of things, okay? So effectively, microorganisms are integrated cyclically. So for example, this guy here can do thiosulfate oxidation coupled to oxygen, or H2S oxidation coupled to nitrate, and H2S is oxidized to elemental sulfur, or pyrite oxidation coupled to nitrate all the way to NO and sulfate. So very versatile organism that can do a number of metabolisms, okay? Now, at a global scale, when you start to put things together, donor and acceptor and so on, you can imagine by any ecosystem being a complex network of electron moving between different species. It's like literally like a circuit, right? And microbes are similar to transistors, in which electrons come in from one chemical species and they are diverted to a different chemical species. And they do this multiple times across multiple type of organisms. So what is a Winogradsky column and why we like it? If you think about our planet is essentially a closed system, with the exception of photons coming from the sun, so energy coming from the sun, a very small amount of matter that we lose to space every day, which is hydrogen and helium, mostly, and then a very, very tiny amount of matter that comes in the form of stardust and asteroids or anything coming from space. But essentially, matter-wise, we are pretty close, and the only input on energy from outside is the photons from our star, right? And this is a closed system where elements are constantly recycled around at the surface of the planet, toward the interior of the planet, between different microbial groups. Like the food we get every day in the form of vegetable and meat and fishes comes from animals that got the food from somewhere else, or come from organisms that can build the biomass from inorganic substrates, like plants do, or primary producers overall. So if you think about Earth is a complex system of interconnected complex system, right? That exchange energy and matter all the time. Have you heard about the Gaia theory? Once in your lifetime? No, never heard of Gaia. Where have you been studying? So Gaia theory is a popular theory that was born in the 70s. There was what we call a cybernetic theory. Cybernetic is a system connected by complex feedbacks, right? And so in the 70s, Lovelock and Margulis set out this idea of our planet as a complex system governed by feedback between biosphere and geosphere. And they went on as saying that our system is actually as hysteresis and as homeostasis, so is resistant to change, which I don't agree with. But the general theory is a bit older, was put forward by a person called Vladimir Vernadsky, which is a Russian biogeochemist, that in 1926 wrote a book called The Biosphere. Now, the biosphere, or the layer of our planet that contains biology, is actually a term created by the geologist US, the come si dice? Austrian geologist, US. Vernadsky took this idea of the layer of our planet where biology is and expanded it, saying that the biosphere is the layer of our planet where biology has an influence. Now, if you're a geologist and you know subduction, you know that this is a process by which plates are pushed deep into the mantle. Now, since life appeared on this planet, we've been pushing into the mantle signature of life in terms of organic carbon, isotopes, and changed in redox chemistry and mineral. Which means that, theoretically speaking, right now in our mantle, where there's absolutely no life because of temperature, pressure, and gradients that are present, we still have sign of life. This is why sometimes people look at inclusion in ultra-deep diamonds, and they're able to claim about the presence of life 4 billion years ago. So according to Vygotsky, life is a geological force that has been shaping our planet. In his idea, life influences a lot of what goes on the planet, well beyond the thin layer that you are at the surface, but is integrated throughout Earth system. It's a very good book. If you never read it, I suggest you do. Vernadsky is very well recognized right now as one of the most influential biogeochemists of our time. And the Goldsmith Society has the Vernadsky Medal awarded to people working in biogeochemistry. But Vernadsky's idea of interconnection of things inspired Gaia theory, inspired our Earth system science, or looking at the Earth system as a complex system of feedbacks, okay? In this context, there's another Russian we need to acknowledge, which is Vygotsky himself. Vygotsky, which was born in 1856, started working in a period of time in which microbiology was just starting off. And one of the biggest problems at the time was to decide if microbes had fixed trait, fixed function and peculiarities that were repeatable over time. So if you can isolate one microbe, ensure that the microbes remain the same generation after generation, then we can classify microbe as we classify plants. So at the time, that was not trivial because you will see this tiny microbe under the microscope, but you didn't know what they were essentially doing. Vygotsky dedicated his lifetime to try to better understand what microbes do in the environment and did a number of groundbreaking discoveries. He discovered the nitrogen cycling, nitrification and denitrification, reduction of nitrogen, oxidation of nitrogen. He discovered that microbes can get energy not just from the sun or organic elements, but also by inorganic redox chemistry. He did that with microbes that can oxidize H2S to elemental sulfur and then elemental sulfur to sulfate. And he realized that microbes in the dark, they can actually produce organic carbon from CO2, similar to plants. He did all that in his lifetime, and these are still the basic concept of microbiology today. He led to the idea of biogeochemical cycle, of the role of bio in geochemistry. And he's the person that started thinking about cycling of planet on planetary scale, which Vernadsky then used on his book. You heard today we have several ways of grow microbes. One of these is by isolating them, right? Taking a complex mixture of microbe and try to isolate a single one. We do this utilizing a system which is called a Petri dish, which is essentially a flat glass container or plastic container in which we can solidify like a gel-like some liquid and then grow microbes on top of it. This idea of isolating the single microbes was developed a long time ago by a person called Koch, Robert Koch, which is a medical microbiologist. Together with his assistant, Petri, they designed this way to isolate microbes to be able to find the disease-causing agent in humans. Koch postulate, which is still today the most important postulate to demonstrate pathogenesis of something, says if somebody's sick and you didn't really like this idea of growing microbes in single cultures, because to him, every microbe that is pulled out of a complex community where there are competition and interaction, and you grow it in a single place where you're giving excess food of every time, doesn't tell you anything of the original microbes in the environment, which is what we've been talking about, right? When we do experiment of manipulation of limited number of factors, we never know what's going on in the real system down there. So he developed a way to grow microbes that doesn't grow single microbes, but grows instead complex gradient of microbes. Now, what you're seeing here are several types of microbes that are layering on top of each other. The colors are just a telltale of what type of. So Winogradsky developed a way to reproduce complex trophic and environmental interaction. Petri developed a way to isolate single bacteria. Now, because medical problems were bigger than environmental problems at the end of the 1800 and 1900, we moved toward single isolation for a very long time. That's what the golden standard in microbiology is. But now that we realize how complex systems are important in systems, there's a comeback to try to think about complex things. So Winogradsky column that we'll see in a second, they've been used a lot as a teaching tool, but not much as a scientific tool in the past 100 years. Now there are several labs working on it, and Feliciana here and my lab have been working on using Winogradsky column as a scientific tool again. So Winogradsky developed this method that he calls the direct method. According to him, every time you want to know what a microbe does in the environment, you need to isolate it fresh from the environment. Because if you reproduce it in the lab, it gets adapted to laboratory condition and is not essentially telling you. Also, you should not make a media out of chemical synthetic compound, but you should do what he calls a soil extract. Basically, dissolving water soil, filter out the particles, or even keep the particles there and use that as a growing media, because that's much more complex than whatever single design you can do. He wrote, in 1923, he wrote, condition of pure culture in artificial environment is never comparable to that in natural environment. One cannot challenge the notion that a microbe cultivated, sheltered from any living competitors, and luxuriously fed become a hothouse culture, and is induced to become in a short period of time a new race that could not be identified with this prototype without special study. So basically he's saying whatever you do in the lab with pure culture or simplified culture is totally useless to interpret the natural environment. Now, we don't say it's totally useless, but we need to recognize that there are big differences between the two, right? And there's actually a very interesting philosophical debate behind the way microbes were isolated by Koch and Pasteur and the letters they sent to each other, attacking each other on the methods that you can read in this paper here. Petri dish versus Winogradsky column, a longitudinal perspective on purity versus diversity in microbiology. It's a very interesting read. Now, you all seen aquarium, right? aquaria are essentially reproduction of marine or freshwater ecosystem. A lot of the cycling in aquaria has to be done by mechanical means. We circulate water, we aerate water, we filter water through a biological filter. We do that because very often our aquarium, we load it with a lot more animals and plants that it can actually sustain normally. So we need to artificially remove waste and recycle elements. But there's a special kind of aquarium called the natural pico or nano aquarium, which are the ones you see here, that normally do not contain any recycling means because they've been thought to contain very few species on their inside that can actually self-sustain each other for a long time. Now, this concept that you might have seen inside actually comes from the Winogradsky column, in which you have these essentially closed to semi-closed system where microbes self-organize and complex geochemical cycle kick in within your system and nutrients are exchanged. So this is just a few of the colors you might create in your Winogradsky column. So for example, these were some that my lab did in 2019 in November. I still have them. They're 5 years old. They're still going. We don't add anything to them except by keeping them facing light. So essentially, they function like our planet functions. It's essentially a closed system with the exception of photons coming in. They went from these on day one when we made them in November of 5 years ago to these. These are the colors that developed. And to a microbiologist, every one of these colors is a telltale of what type of metabolism microbes are doing. Are they using H2S? Are they using iron? Are they using light without oxygen? Are they producing oxygen? Are they autotrophs, heterotrophs, and so on? So, and the gradients we see here are very similar to the gradients I mentioned in the environment. Look at this rock here. This is a crystalline rock. And look, that's the surface. And look at the gradient here with the green and the brown color sitting right there. Those are the endolytic microbes I was telling you about. They grow just below the surface of the rock. And there, they change completely in the mineralogy, the flux of elements, and the interaction. Yes? That's biological. You were like, oh, what a strange layer of copper sulfate I have in my rock. I often wonder how many times geologists collected carbonates around to do the isotope, and they get negative isotopes, but not because of the carbonate, because of all the organic carbon they have inside that they don't realize. So that might be part of some things, depending on the techniques that are used. And this is another one. This is the same video we were looking before. This is not an endolytic, this is a superficial, but it's bioprecipitating calcite through its layer. So there is a significant portion of calcium carbonate in this biofilm, but you can see the different layer and the different colors. And each one of those represents a population of microbes that has a set metabolism, often redox, and is exchanging things above and below itself. So Winogradsky column, what happens? You start like this, okay? We're gonna mix some mud we collected from a geothermal place very nearby in the Campi Flegrei here. We're gonna mix it with a little bit of water and with some nutrients. We'll discuss what we will give them, and you are free to decide. And we're gonna essentially create an homogeneous mixture of mud and nutrients. And then we're gonna layer it on top with some water to avoid oxygen to get directly in. Then we're gonna close the system. So we start essentially from an homogeneous mixture, okay? What happened in this mixture? Well, to start, we're gonna get a gradient of oxygen, right? Because you have compaction of the thin sediments at the bottom. So you have a gradient of oxygen going down. At the same time, if you're using mud that comes from a geothermal system that was essentially anaerobic, you get other gradients going, and we'll see in a second. So what happens at the top? Well, you have water, you give it light, you most probably have microalgae. They like to do oxygenic photosynthesis. So they take CO2 and they make sugar while oxidizing water to oxygen, this is what plants and animals do. That will be the norm that happens at the top. At the same time, you have produced sugar and oxygen, so there are heterotrophs. They will burn the sugar with the oxygen to make CO2 and water. So this is already a closed loop, right? Primary producer, secondary producer. Plants, herbivores, if you want, on land. In this case is oxygenic phototrophs and aerobic heterotrophs. At the same time, at the bottom, you get gradients of H2S or H2, depending what your starting MAD was. You start to get from the more compacted anoxic layer, gases will rise to the top, but they will make oxygen. So you get a gradient, right, from reduced to more oxidized. At the bottom, why you start to get hydrogen in a system like this? Well, if you're anaerobic and your MAD is loaded with organic carbon like ours is right now, you get the famous fermenters. The microbes that break down organic carbon without any acceptor because there's no oxygen. Oxygen is there at the start when you mix, but microbes will consume it very, very fast, right? So unless it's continuously produced by oxygenic phototrophs, there's going to be zero oxygen at the bottom. So people will start to produce hydrogen. Hydrogen is light, starts to go up. We get a first gradient. The hydrogen can be used by microbes to do sulfate reduction. So suddenly, microbes are doing sulfide down there using the sulfate that is present. Well, if your initial sample has sulfate, right? Or if you provide sulfate to your system. If you do sulfate reduction, you get H2S. But you also have iron most probably in your sense. So microbe can either do photo iron oxidation or iron reduction. If you have iron 3, you are in the anaerobic layer. And then eventually you can have photo or non-photo sulfur oxidation, because you get also a gradient of sulfide now going up. A sulfide might emit oxygen somewhere, a microbe might do that. So suddenly we have 2, 3, 4, 5, 6 elements that are exchanged up and down because of this natural gradient that are created essentially by gravity. Compaction, water stays at the top, sediment compact at the bottom, gases rise, things diffuse down. So we're starting with an homogeneous structure, but we are not in vacuum in space, so there is a least gravity, and gravity is going to change the gradients within our system. I think they're mainly selected by the different layer because swarming centimeter doesn't work in sediments. At the bottom, you may even get methanogenesis if you are compacted enough and anaerobic enough. So you start to get methane as well that then comes up and can be oxidized at the expenses of sulfate, elemental sulfur, nitrate, iron 3, or oxygen. And you go from this to this. And these are exactly the layers. So when this changes into this, you see the green. The green are the oxygenic phototrophs. And then you see a layer of heterotrophy here. And these guys are actually photosulfur oxidizers. And down here, that's probably amorphous elemental sulfur, the white one you see there. And you're getting sulfur being precipitated by sulfide oxidation that is coming from the bottom. They become dark black because conditions are highly reducing. Yes? Only an oxygenic phototrophs at the top. So for some reason, the system remained so reducing and anoxic, and there was so much H2S coming from the bottom that things didn't kick in. What happens that even if you start from the same condition like we will do today, you can change the condition between the columns. So one of you might select to mix the mud, add it, close it. I don't want to touch, I don't want to add anything external. Other people might want to play with your system. I'll give you an example. We discussed the way electron acceptors are used going down based on amount of energy, right? When you're in sediment, oxygen disappears first, and then nitrate, manganese, iron, sulfate, CO2 as you go deeper. They give you less energy, so they're used after, but they get used as electron acceptor. Every one of those compounds needs a special metal to be used. We discussed this as oxygen needs copper and so on. So just by changing one metal you provide to your column, you might change the stratification of your layers. And then if you provide sulfate as sulfate ions to a column where there are no sulfate, we were discussing yesterday worst case scenario in underground hydrogen storage. We give sulfate, lactate, acetate, and sugar, and let's see how things change, even if the inoculum is the same. So what we will be doing today is essentially playing with this. So electron donor, electron acceptor, a carbon source. We start from that. And then additional things, cofactor, trace elements, and vitamins. So on the menu today, you have carbon, Inorganic as CO2, that's provided by the air and the waters that are there, or eggshell for calcium carbonate. You have organic matter that is already present there, but you can add cellulose, or you can add sugar, table sugar, the one we use for coffee. Then we have nitrogen sources you can add. We have fertilizers with us. So you can add, for example, some fertilizers which provide nitrate, phosphate, and ammonia. Or you may decide to add electrolytes like Gatorade or stuff you drink. Those contain also minerals and fertilizers. You may add phosphorus. You may decide to add trace element, vitamins. You can make it in the dark or in the light. So you can mix these things as you want, right? So what we will do, essentially, is we will mix the mud we have here for you, and it's going to be only mud mixed. Then you come up, you take one of these flasks, which are semi-permeable membranes. So to bring it back home in your luggage, please seal this with some tape. They don't leak water out, but it's better to put. They're only for gas exchange, but it's better if you cap it completely. And then what I want you to do is to come to your vials here, and then you mark a height that is around here, 150. And you're gonna take one of the plastic container we have, which are plastic glass to drink. You're gonna put some mud inside there, and then you're gonna add whatever ingredients you like. You wanna give it sugar, you wanna give it egg, you wanna give it both. We go back to this list, and you select a few ingredients that we have. You can put everything. You can put nothing. You can play in your mind if you want to provide excess sulfur or excess phosphorus or excess ammonia, whatever you want. You make your unique mix of these things, and then you pour inside the column up to this level, your mud. You fill it in with water about up to here, to the top, and then you close it, label it, date, name, ingredients, and this is done. Now, you go back home or your office or wherever, you find a nice window, you just sit it in front of the window, facing the window with one side, and you wait. And after 2 days, 3 days, mud is going to start to compact and settle. After a couple, 2, 3 weeks, things will settle really, really well, and you might already see some gradients creating. Usually within 1 to 2 months, you get full colors going on that tell you what the gradients are. So comparison between the different columns you will do might tell you why some metabolism are more abundant somewhere than the other, depending on what you added with your column, right? What you hyper-stimulated as far as the community goes. Any question before we start? Yes. Well, we, for example, right now, Feliciana is looking for reproducibility of the column. Once you get a gradient established, if you repeat the exact same column and you re-inoculate, how many times looks exactly the same? How variable that is. And then can we manipulate the gradients controlling, for example, only one of the parameters? So if we change only the amount of sugar or copper or phosphate, how much are we changing this? And we don't do this just by color, but we extract DNA from the column and sequence and see in composition space how much the community changes between replicates, between repetition, and over time. Well, theoretically speaking, if your inocula is homogeneous, your starting mud is homogeneous, which is not always the case because, for example, the mud has been sitting there for a day and a half, right? So it's already stratified with thinner mud at the top and thicker at the bottom. Just changing the granulometry of your column will dictate how much the oxygen is penetrating down. The finer the sediments, the less penetration and more anaerobic and reducing condition we'll have at the bottom. The coarser the things you pick, other way around. If you leave there a small branch of wood that goes vertical down, that's a source of lignin and cellulose, but might also be a disturbance conduit for flow to move across the layer, right? Because there's a discontinuity where gases can use to move faster. So assuming you have a completely homogeneous sample between the two of you, and you use the exact same condition, what we are seeing in the lab is that conditions are pretty much reproducible. So within a certain range, the type of color you get and the layering is very similar. Then, of course, there is plus or minus 1 millimeter, higher, lower, and so on. But we have replicates that Feliciana has been doing where we see that they're pretty consistent in the way they respond. Next question. We are using these systems. So first to establish the variability and manipulative effects. One thing we do that we will not do here is that to increase this gradient, to make this gradient stronger, we are actually putting at the bottom here a plug of sodium sulfide to increase the amount of sulfide coming from the bottom, which better simulates geothermal system in which you have a deep source of reduced sulfur with the volcanoes, right? But we are trying to assess the variability of these things. At the same time, we're trying to assess how reproducible they are. We are using them to isolate new member of the community, because when you start from the mud, your microbe may be so rare that you never get it isolated. This is a complex mixture enrichment where then you can pick out one single type of microbes and isolate it in the environment. And we started trying to do some manipulation. Our interest is on trace element. So we started providing to the same column, reproduced over time, different set of trace elements to see if we can overstimulate certain metabolism, specifically by trace element addition or depletion. We are in the middle of our PhD on this. But I feel some of this complex system reproduction will be crucial also to understand behavior in subsurface, because for as homogeneous you want to start, there are natural gradients that create within your experiment, especially if the time scales are wrong, right? You shake, you do an experiment that is 2 hours, okay. You shake, you do a sitting experiment for 6 months, you're going to get gradients in your experiment. There's nothing homogeneous there. Any other question? And actually, some of these gradients that are born in a container like this are very difficult to predict and then model, because you would assume that anything you run in an experiment like this is homogeneous. So you're modeling it as an homogeneous system most of the time. I put water, sand in a known amount, I mix it. The redox gradient that are created here are non-trivial to reproduce unless you're aware of it and you specifically plan. Possiamo